In this third video for Unit 4, we're going to be talking about synovial fluid and the serous fluids. Okay, so some physiology, first talking about synovial fluid, also known as joint fluid. It's a viscous liquid formed as an ultrafiltrate of plasma across the synovial membrane, and it's found in the cavities of the movable joints. The purpose of synovial fluid is to reduce friction between the bones during joint movement and to provide nutrients for articular cartilage. Testing of synovial fluid is frequently done to determine the cause of arthritis, and some of the common tests performed on synovial fluid include a white cell count, differential, gram stain, culture, crystal examination, glucose, protein, and uric acid. So we'll be talking about each one of those tests. So specimen handling and collection. It's collected by a needle aspiration. That procedure is called an arthrocentesis, and the amount of fluid collected depends on the joint that it's collected from and the extent of the buildup of fluid. So if there's only a little tiny amount of fluid build up, then you can expect only, a small sample. Normal synovial fluid should not clot, but diseased joints can produce fluid containing fibrinogen and will clot. The aspirate should be separated into the appropriate containers for testing that is to be performed. So for cell counts, you want some of the sample put into an anticoagulant tube, and liquid EDTA tubes are better than powder, and that's just in order to prevent artifacts during the crystal examination. You want a sterile container for microbiology testing and a plain red tube without the anticoagulant for your chemistry and crystal testing. And that's really true, that scenario. All of this is kind of true for really any body fluid that you would collect. So it would be for the synovial fluid and the serous fluids that we're going to talk about next. Okay, normal color of a synovial fluid is colorless to pale yellow. That pale yellow is sometimes referred to as straw, and with no clots. It can be darker in cases of effusions, which are accumulations of fluid. They can even have a green tinge in cases of bacterial infections. They can be tinged with blood if the procedure, if the arthrocentesis is a traumatic aspiration. And then turbidity in the sample is associated with white cells, but it can also be due to debris or fibrin. It can be milky when crystals are present. The viscosity in synovial fluid comes from the polymerization of hyaluronic acid, and arthritis affects the production of hyaluronic acid, which would cause the increased viscosity. There are several tests that measure this synovial fluid's viscosity. One is called a mucin clot test, and there is a video in Moodle that shows the mucin clot test, so make sure you watch that one. But that one is not a routinely performed test much anymore. Another test determines the specimen's ability to form a string from the tip of a syringe. If synovial fluids are too viscous, they would have to be treated with hyaluronidase, which breaks down the hyaluronic acid in the sample, and it'll make the fluid less viscous so it can be tested. If you've got a very viscous fluid and you try to run that sample on your automated hematology or chemistry analyzers, it can gunk up the machine. So you want to make sure to use that hyaluronidase, which is a powder. You just add like a very, very tiny amount to the fluid, and it'll make it much thinner and able to test. Okay, for your cell counts, white cell count is the most common. Red cell count is not as commonly requested. The counts should be performed as soon as possible after you receive the specimen in order to prevent cellular disintegration. The counts can be done on the hemocytometer, but many automated counters now perform the cell counts. So that's your automated hematology analyzer. As long as that instrument has been validated to run synovial fluids, you can run the sample on your hematology analyzer. But just remember, if the fluid is very viscous, you would need to treat it with hyaluronidase to decrease that viscosity so that it's not going to clog up the machine's aperture. White cell counts of less than 200 are considered normal. And if it's necessary to dilute the sample, you can use normal saline for that. For the differential count, it should be performed on a cytospun slide, same for your gram stain. Most white cells seen in synovial fluid should be mononuclear cells, so that would be like your monos and your lymphs. Neutrophils should account for less than 25% of the cells counted. Lymphs should be less than 15%. Increased neutrophils could indicate a septic condition. Increased lymphs could indicate a non-septic inflammatory process. And a variety of other cells can also be seen. There's a chart in the book that talks about those different kinds of cells that can be seen in the conditions that they might be seen in. For your chemistry testing, the most frequently done test on synovial fluid is glucose, and a blood glucose should be performed at the same time as a comparison. Synovial fluid glucose should not be more than 10 milligrams per deciliter lower than the blood value. Very decreased glucose levels could indicate inflammatory or septic disorders. Some other chemistry tests that can be done on synovial fluid are your total protein and uric acid. Normal synovial fluid should contain very little protein, so you might see increased levels in inflammatory and hemorrhagic disorders. Elevated uric acid levels are seen in gout and can be used to confirm gout when crystals cannot be seen in the synovial fluid. And then lactate levels can also be used to monitor rheumatoid arthritis. For microbiology, your gram stains and cultures are used to detect bacterial infections, but fungal, tubercular, and viral infections can also occur in the synovial fluid. Some common organisms recovered from synovial fluid bacterial infections include Staphylococcus, Streptococcus, Haemophilus influenza, and Neisseria gonorrhea. For your serologic testing, this would be done on serum to test for rheumatoid arthritis and lupus, which are causes of joint inflammation. Lyme disease can also cause arthritis, so the serum would be tested for antibodies demonstrated in each of those particular diseases. Also, a CRP and a fibrinogen test can be performed to test the extent of the inflammation involved. The information that you see on the slide here about the disorders is as much as we'll go into these classifications of disorders. So you've got non-inflammatory, which would be degenerative. Osteoarthritis would be an example. Inflammatory conditions would be immunologic. Examples of those would be lupus, rheumatoid arthritis, and Lyme's disease. Septic classification would be due to a microbial infection, and then hemorrhagic would be due to trauma, tumors, or some type of coagulation deficiency. Then the crystal identification. So this is very important and common test done on synovial fluid for evaluation of the cause of arthritis. Crystal formation can be due to metabolic disorders and decreased renal function, among other causes, and they can be chronic or acute. So some of the different types of crystals that can be found, the main types of crystals seen in synovial fluid are going to be monosodium urate, otherwise known as uric acid, sometimes referred to just as MSU. So those crystals are seen in gout, calcium pyrophosphate dehydrate, or CPPD, and cholesterol crystals can be seen in pseudogout. Frequent causes of gout include impaired metabolism of purines, increased consumption of high purine content foods, alcohol, and fructose. Also, chemotherapy treatment for leukemias and decreased renal excretion of uric acid. Frequent causes of pseudogout could be degenerative arthritis and endocrine disorders. Other crystals can also be present. We're not going to go into those as much, but you can read the slides and look at the charts in the book if you want to learn about some of those additional types of crystals. So for the slide examination, this should be performed as soon as possible after receiving the specimen because crystals can be affected by changes in pH and temperature. Also, the crystals are often seen intracellularly and the cells can deteriorate. The initial exam is done as a wet prep with 1 drop of fluid on the slide with a cover slip and examined under low light and high light. And then it should also be examined under polarized light with a red compensator for identification of the crystal. MSU crystals are usually needle-shaped. CPPD crystals are usually rhombic-shaped, square, or they can also be short rods. The presence of crystals is first done under regular polarized light, so both your MSU and your CPPD crystals will polarize, but your MSU crystals are more birefringent and they will be brighter than the CPPD crystals on a dark background. To confirm the actual identification of the crystal, you would then use a red compensated polarized light. So you've got a red compensator, which is just kind of a filter that you can put on your bright field microscope. It's placed between the crystal or the sample, the slide, and the light source to produce a red background. And that compensator separates the light into slow and fast moving vibrations. Then the crystals should be aligned with the slow moving vibration and examined for the color that they produce. And I'm going to skip ahead to this slide here. So this is as if you are looking down into the scope. You've got this filter, this compensator that's been added to your microscope that's got a little handle on it. So when that handle is turned to your right, that's considered the slow vibration. If that handle is facing towards you, that's considered to be turned to the fast vibration. So based on that, your MSU crystals will be yellow, which is referred to as negative birefringence, under the red compensator when parallel to the compensator when it's turned to the slow vibration. So again, that's your handle to the right. Your MSU crystals are yellow when they are parallel with the handle. And then the CPPD would be opposite. They're going to be perpendicular. And then that's considered gout. If you identify these crystals, CPPD crystals will be blue under the red compensator, which is referred to as positive birefringence, when the crystals are perpendicular to the compensator when turned to the slow vibration. So that's called pseudogout. And then when the compensator is turned to the fast vibration, the colors are going to be reversed. So at that point, MSU would be blue and perpendicular, and your CPPD would be yellow and parallel. Okay, and you can just review that slide. There's some your normal synovial fluid values for the different tests that we perform. So just take a look at that. Okay, this next little lecture is going to be on the serous fluids. Okay, serous fluid is the fluid between the membranes in the pleural or lungs, the pleural space, the pericardial space, which is the space that surrounds the heart, and the peritoneal space, which is what surrounds the abdomen. Provides lubrication to prevent friction between the membranes that occurs with the movement of organs. So for instance, the contraction and expansion of the lungs. It's formed as an ultrafiltrate of plasma. There's a constant exchange of serous fluid between the membranes in order to maintain a constant volume between the membranes. When that mechanism is disrupted, the result is an increase in the amount of fluid between the membranes, which is called an effusion. Causes of effusions include congestive heart failure, hypoproteinemia, inflammation and infection, and lymphatic obstruction, such as tumors. These fluids are collected by needle aspirations. So the procedures are referred to as a thoracentesis, which is the collection of the pleural fluid, pericardiocentesis, which is a collection of pericardial fluid, and paracentesis, which is the collection of the peritoneal fluid. That peritoneal fluid you will also sometimes hear referred to as an ascites, which is spelled ASCITES. CITES. So if you see that word, it's referring to peritoneal fluid. Specimens should be collected in the appropriate tubes depending on the testing that's to be performed. So just like we mentioned for the synovial fluid, you've got your EDTA tube for your hematology testing, a clotted or heparin tube for your micro, and cytology, a clotted or heparin tube for chemistry and serology. And specimens that have a pH test ordered should be kept on ice. For microbiology testing, you would want to centrifuge the specimen for the gram stain, and that's true for the differential also. And for the chemistry test, you usually have blood drawn at the same time for comparison. Okay, transudates versus exudates. This is another topic for which there is a video in Moodle, so make sure you take a look at it. The cause of the effusion is characterized as either a transudate or an exudate, which is helpful in determining the need for further testing. Many tests can be used to differentiate between a transudate and an exudate, but the most helpful tests are the fluid to blood ratios of protein and lactate dehydrogenase. Transudates are effusions that are caused by systemic disorders that disrupt the production and regulation of fluid that is filtered and reabsorbed between the membranes. So examples of those types of conditions would be congestive heart failure and nephrotic syndrome. Exudates are produced by conditions that directly involve and cause damage to the membranes of the cavity. So examples of those conditions would be infection and malignancy. Exudates are the ones that are usually going to require further testing. Transudates usually do not. And there's a slide here that shows some of the main differences in testing between transudate and an exudate. So the doctor would examine these results, determine whether we're dealing with a transudate or an exudate, and then would determine the need for additional testing. Okay, so classification as a transudate or an exudate, appearance of the fluid, cell count, differential, chemistry, micro, cytology testing is all normal and can vary slightly on the origin of the fluid. Okay, so now we're talking about the pleural fluid. So again, this is from the pleural cavity around the lungs. So in addition to the tests routinely performed on serous fluids, pleural fluid cholesterol and fluid to serum cholesterol ratios and the pleural fluid to serum total bilirubin ratios are also helpful. Normal appearance should be clear and pale yellow. A turbid or white sample indicates the presence of white cells related to infection. A milky sample is referred to as chylous, so it could contain chylous material, which are large amounts of triglycerides from a thoracic duct leakage, or from pseudochylous material, which means that it would contain large amounts of cholesterol, which is produced with chronic inflammations. Blood in the pleural fluid can indicate a hemothorax, which is an accumulation of blood in the pleural cavity caused by some type of traumatic injury, or hemorrhagic effusion, which is common in tuberculosis and malignancy. There are other colors possible. Again, there's a chart in the book that you can reference if you want to look at some of those other colors that are possible. Your hematology testing, so you would have your cell counts. On the differential, you can see macrophages, neutrophils, lymphs, eosinophils, mesothelial cells, plasma cells, and malignant cells. Increased neutrophils would indicate a bacterial infection. Increased lymphs could indicate tuberculosis, viral infections, malignancy, and autoimmune disorders such as rheumatoid arthritis and lupus. Increased eosinophils can be associated with trauma to the pleural cavity, allergic reactions, and parasitic infections. Mesothelial cells are just your normal membrane lining cells, and they have what is called a fried egg appearance. And a lack of these cells would be significant and would indicate tuberculosis infection. Malignant cells can be hard to differentiate from other normal cells, but will usually have a very irregular appearance, and special stains and flow cytometry can be used for positive identification of tumor cells. For your chemistry tests, so a glucose pH, adenosine deaminase, or ADA, and amylase are the most common tests performed. Triglycerides and lactate can also be done. Lactate is produced in muscle cells and red cells and forms when the body breaks down carbohydrates for energy when oxygen levels are low. So it would be increased in infection or disease, but it can also be increased after intense exercise. Decreased glucose levels would be seen in tuberculosis, rheumatoid inflammation, and infections. Lactate levels are elevated in bacterial infections. For your micro and serology testing, we're going to do gram stains cultures, AFB stains, mycobacterial cultures. Most common bacterial infections are with Staph aureus, the Enterobacteriaceae, anaerobes, and Mycobacterium tuberculosis. Serology testing can be used to differentiate effusions of immunologic origin from non-inflammatory processes. And they have tests for anti-nuclear antibody, or ANA, which could be an indication of lupus, and rheumatoid factor. So those are some of the most frequently performed serology tests. Also, tumor marker tests can be performed to determine malignancy. Okay, and here's just a kind of a recap of some of the chemical testing that can be done on pleural fluid and what the indications for those would be. Okay, next is pericardial fluid. So there's usually only a very small amount of this fluid present. Effusions are primarily the result of exudates due to changes in the membrane permeability due to infection, which is referred to as pericarditis. Could be also due to malignancy or trauma. Transudates are due to metabolic disorders, hypothyroidism, uremia, which is urea in the blood. It's associated with fluid, electrolyte, and hormone imbalances and metabolic abnormalities, and can also be in immune disorders. Again, the normal appearance of the fluid should be clear to pale yellow. Turbid would indicate infections and malignancy. And malignancies can also sometimes be streaked with blood. Grossly bloody specimens are associated with cardiac puncture and misuse of anticoagulant medications. A milky fluid would indicate that chylous or pseudocylous materials are present. Okay, some of the lab tests that we perform. The testing that we're going to do is primarily going to be to determine if the fluid is a transudate or an exudate. So this includes the fluid to serum protein and LD ratios. White cell counts are not of much clinical value except when you have values greater than 1,000 per microliter with lots of neutrophils, which indicates a bacterial endocarditis. The cytology exam would look for malignant cells, tumor markers. Bacterial cultures and gram stains are common, and infections, bacterial infections in the pericardial fluid would be common with strep and staph species. Effusions of tubercular origin are increasing as a result of AIDS, so AFB stains are becoming more commonly ordered on this fluid as well. Here's kind of your recap on the testing that can be done for pericardial fluid and what the significance is. Okay, last is the peritoneal fluid. So accumulation of fluid between the peritoneal membranes, which is in the abdomen. And remember, that's called an ascites or peritoneal effusion. It's collected by means of a peritoneal lavage where saline is introduced into the abdominal cavity and then aspirated. Red cell counts greater than 100,000 per milliliter are indicative of blunt trauma injuries. Other causes of these effusions could be due to hepatic disorders, bacterial infections, and malignancy. Transudates versus exudates in peritoneal fluid. So it's more difficult to determine than with pleural and pericardial fluids. And it's usually going to be done by measuring the serum and fluid albumin levels. Appearance, once again, the fluid should be clear and pale yellow is normal. Exudates are turbid in bacterial and fungal infections. You can have a green to brown fluid that would indicate the presence of bile. Blood streaked could be either due to some type of trauma or in cases of tuberculosis, intestinal disorders, and malignancy. Your chylus and pseudochylus could indicate trauma or lymphatic vessel blockage. So some of the lab tests that we would perform, your normal white cell count should be less than 350 cells per microliter. The count's going to be higher in bacterial infection and cirrhosis. So to distinguish between the two, you could do a differential. Greater than 50% neutrophils indicates infection. Lymphs are going to be predominant in tuberculosis. Cell exam is important for detecting malignant cells to identify tumors. Some of the other cells that you can see in this type of fluid are macrophages, white cells, mesothelial cells, which again are just membrane lining cells. And then microorganisms can also be seen, such as bacteria, yeast, Toxoplasma gondii. Some of the chemistry testing that we would do, glucose, amylase, and alkaline phosphatase are the most common. The glucose is below serum levels in bacterial and tubercular peritonitis and in malignancy. Amylase can help with the determination of pancreatitis. Elevated ALP is diagnostic of GI perforations. Your BUN and creatinine can be ordered if accidental puncture of the bladder is suspected. And then bilirubin is measured when leakage of bile into the peritoneum is suspected. And then finally, microbiology testing, our gram stains and culture for aerobic and anaerobic organisms, and AFB stains and cultures, and adenosine deaminase could also be ordered, which would check for a genetic deficiency of an enzyme. So that's the end of this little lecture on the synovial and serous fluids. We'll pick up in the next lecture with amniotic fluid and fecal specimens.